Hi everyone, my name is Ming Vu and I am a master's student in biotechnology at Brock University. My research focuses on grapevine viruses. This has become a major problem for many grape-growing regions in the world because grapevine viruses have spread globally due to global trading. My first project focused on a grapevine virus that has wreaked havoc in North America, namely the grapevine red blotch virus. My goal was to see if this virus is being spread in the Niagara vineyards of the Ontario region. To do this, I deployed virus-free, clean sentinel vines onto an infected field and then test them to see if they are being infected from the surrounding. Luckily for us, my results show that there's no sign of red blotch being spread. However, 70% of all sentinel vine that we deploy got infected with another virus called grapevine Pinot Gris virus. This is very concerning because we only exposed them for 4 months, so the spreading speed is staggering. So that led us to the second project that I've done. After a lot of investigation, we have found that the source of this virus in North America is most likely from importing unclean material from France, and but also most of the Pinot Gris in North America are asymptomatic, meaning that the damage will be minimal. My research have highlighted the importance of clean plant material and how we need our own North American source of reliable, clean, virus-free material to move forward. As for me, I would like to continue to do more research, find more results, contributing more to the sustainability and profitability of this industry. So thank you for joining me today, and remember to drink some wine. Thank you so much.
My name is Kyle Friedman, and I'm currently a PhD student in horticultural sciences at North Carolina State University. So the main research question that we're trying to address is if we can condition young grapevines for increased and early flowering and fruiting capacity. So we're looking at the manipulation of different environmental factors like light intensity to see if we can have earlier flowering and increase the fruiting capacity of a vine so that when we transplant these vines in the field, we can have an earlier crop than what you would normally have in typical grape production. The highlights of my research have really been a combination of working in a controlled environment as well as being able to bring that research into the vineyard and sort of have that combination of experiences. There's just so many different states that are really getting into grape production that traditionally haven't produced grapes or made wine and really experience a lot of different challenges that benefit from some novel research. So I think the grape industry is experiencing this sort of renaissance of trying to figure out what cultivars work in specific regions of North America that traditionally have not been common grape growing regions. So I share my research in a couple different ways. We try and incorporate growers into our research directly. Working with them, we've been able to share what we're doing and some of those results. And then also conferences like the American Society of Enology and Viticulture Eastern Section that we're a part of, I present at annually, other conferences internationally. One of the most impactful results of our research that we found is that as we increased supplemental light intensity, we had an increase in the total clusters per vine, particularly for the medium light intensity, which was the highest number of clusters. And what this means for us is that we can refine production systems that intentionally condition grapevines in the nursery for higher fruiting capacity and ultimately higher yield, which can produce fruit in the first year in the vineyard. So after my PhD, I'll be looking at professorship positions in the United States, so predominantly focused on viticulture. I'd like to stay in the industry and both continue some of the current research I've been doing in my PhD as well as other topics that can really support growers and the growing grape and wine industry in North America.
My name is Elia Castellanos and I'm a master's student at the University of British Columbia. My research project is part of a collaboration with the Okanagan Crush Pet Winery in the Okanagan Valley, BC. The main objective of my research is to test indigenous Saccharomyces cerevisiae strains that have been isolated from OCP vineyards and winery environment and that have been confirmed to be genetically distinct from those strains commercially available, and assess their potential as wine fermentation starter cultures. To do so, we have selected 6 strains and performed lab-scale fermentation in Pinot Noir and Pinot Gris. After evaluating the fermentation performance as well as primary and secondary metabolite production, 2 strains were selected to carry out pilot-scale fermentations at the winery. These wines will be evaluated using liquid chromatography and gas chromatography to quantify major metabolites and volatile aroma compounds, and this data will be compared to sensory analysis data, which will help us describe the aromatic profile of the wines produced with the indigenous strains. My favorite part of this project is having the opportunity to work side by side with the wine industry and address one of the many challenges that they face during the winemaking process. Having yeast strains that are well adapted to the environment in which fermentation will be taking place makes a great contribution to create high quality, distinct wines. This differentiation can help winemakers stand out their product in the international market, benefiting from those characteristics specific to their wine regions. I want to thank the American Wine Society Educational Foundation for selecting me as one of the 2023 AWSEF scholarship recipients. Thank you for supporting my research and my education.
Hello, my name is Megan Mershon, and I am currently pursuing my master's degree in food science and technology in Dr. Amanda Stewart's lab at Virginia Tech. My focus is on wine and how fertilization with nitrogen in the vineyard impacts wine chemistry and sensory properties. Nitrogen is an essential nutrient to grapevine growth, grape quality, and wine fermentation. Yeast used to ferment wine require nitrogen to grow and reproduce. The quantity and qualities of nitrogen sources in a grape juice prior to fermentation can impact the outcome of a final wine. Because of nitrogen's importance in the winemaking process, we are asking the question, how does adding nitrogen in the vineyard or the winery impact wine quality? We are working with commercial growers in Virginia to grow grapes with different levels of nitrogen fertilization. From those vineyards, we are harvesting grapes from each treatment to make wine. We then are performing a variety of analyses to understand what the chemical differences are between the wines. We are then asking consumers to taste our wines to determine if there are differences between the treatments. In the coming year, we are going to be evaluating how nitrogen addition by fertilization in the vineyard might impact wine quality differently from nitrogen addition in the winery. The goal of this research is to serve grape growers and winemakers of North America in better understanding their product. If we can understand the impact that nitrogen has on the quality of the wine they are producing, they can better produce grapes and wine across the country, the continent, and the world. That is what I'm passionate about with this research. I want to help growers and winemakers thrive in an ever-changing environment. The East Coast provides a challenge to grape growers, and I am excited about contributing to the body of knowledge they have in order to help winemakers create wines that are competitive on a world stage. I would like to thank the American Wine Society Educational Foundation for investing in me, my research, and my future. Thank you, and cheers to improving the wine industry one glass at a time.
Howdy from Texas. My name is Michael Cook, a viticulturist with the Texas A&M AgriLife Extension Service with the Department of Horticultural Sciences at Texas A&M University. I'm also a PhD student within the department with an applied focus on viticulture that also includes an enology component. Before I talk a little bit about my research and what I do and the fact that Texas actually has an exciting wine industry, I wanted to first of all thank the American Wine Society for your commitment towards the American wine industry. I know it's unwavering. Cannot thank you enough for that support, both supporting our industry as a whole, but also the students, the next viticulturists and enologists and researchers for the United States. So it's a privilege and an honor to be a recipient of the American Wine Society. I want to thank you for that support and helping me further my education so that I can give back to our industry. So you may be wondering who this Texan is, and does Texas even have a wine industry? We absolutely do. We've been growing grapes since the 1650s. It's been a long, arduous battle, and one of those challenges relates to water, and also as it relates to red wine production with high pH wines, over 4. So we know irrigation is important. We know that we have to irrigate here in Texas, but how often, how frequently, that's going to be dependent on many factors. And so my PhD research is looking at the effects of irrigation regime in providing enough water to the vines with water conservation in mind. also having an effect on potassium uptake and hoping to reduce the amount of potassium that's shunted into the fruit. So we're looking at the effects of irrigation on wine quality, wine stability, but also grapevine resiliency. So again, I want to thank the American Wine Society for this opportunity in supporting me in my continued education and commitment to not only the American wine industry, but the Texas wine industry in particular, and my growers that I work with in my region, but also across the state.
Hi, my name is Meredith Persico, and I'm a graduate student at Penn State University entering the final year of my PhD in horticulture. First, I would like to thank the AWSEF Scholarship Committee for selecting me for this award. This award was funded by the Carroll County, Maryland AWS chapter and G. Hamilton Mowbray in memory of Carmen Blosfarin and in honor of Richard Blosfarin. I'm so honored to be recognized for my graduate research. My doctoral dissertation is on grapevine cold tolerance. So that's how grapevines cold acclimate in the fall to prepare for dormancy, and then in the spring, how we can help growers mitigate spring freeze damage. Over the past few years, I conducted a series of experiments looking at how increased temperatures in late summer and fall might affect grapevine's ability to cold acclimate and get ready for cold temperatures in the winter. Over the next year, I'll be analyzing my data and writing up my results. I hope these results will be useful to grape breeders to breed grapes for improved cold tolerance. I also hope these results will help practitioners better prepare to grow grapes in the face of climate change. After graduate school, I hope to stay in the industry as a research scientist, continuing to help solve challenges in grape production. Thank you again to AWS and the AWSEF Scholarship Committee for selecting me for this award. Cheers.
Hello, my name is Alex Kopinski. I am a second-year master's student in food science and technology at Iowa State University. There wasn't a grapevine in sight when I grew up in northern Minnesota, so I was introduced to the world of wine when I studied abroad in France. My desire to dig deeper into the science of wine led me to research focused on cold-hardy grape cultivars, such as Marquette and Frontenac, that grow in my home region of the upper Midwest. One of the primary challenges associated with red wines made from these grapes is lower tannin concentration than red wines made from popular Vitis vinifera cultivars, such as Cabernet Sauvignon and Pinot Noir. Tannins are related to wine quality and are important because they are responsible for the mouth-drying effect of red wines and in stable color formation and wine aging. In an effort to address the low tannin challenge and increase the level of tannin in Marquette red wines, my research is looking at the effect of two sprays that are applied to the vine canopy on tannin content. The first spray is prohexadione calcium, which is a plant growth regulator that limits cell expansion during berry growth, thus concentrating tannins in the grapes. The second spray is phenylalanine, which is an important nutrient for tannin biosynthesis. My hypothesis is that by giving the vine extra building blocks, it will make more tannin that will end up in the final wine. Because tannin in wine comes from the skins and seeds of grapes, I am examining the phenolic content in those different berry tissues, along with the finished wines. To do this, I use analytical techniques including high-performance liquid chromatography and UV visible spectrophotometry. The first year of this project has shown promise, and I am excited to analyze the results from the second year this fall. Hopefully, my research will provide wine growers using cold-hardy hybrids a tool to increase tannin concentration and thus the quality of their red wines. Thank you to the American Wine Society Educational Foundation and the Meadowlands, New Jersey AWS chapter for selecting me to receive this scholarship and for giving me the opportunity to share my research with a wider audience. There's always more to learn in the world of wine, and I'm grateful for the support. Thank you. Cheers.
Hello, my name is Nadine, and I'm grateful to be receiving a scholarship from the American Wine Society. I'm currently pursuing my master's degree in biotechnology supervised by Dr. Deborah Ingalls here at Brock University, located in Niagara, Ontario. About a year ago, I had the opportunity to volunteer for the International Cool Climate Wine Symposium. It was here that I got to see just how big the wine industry is and how interconnected we are internationally. I also had the amazing opportunity to volunteer in a Chardonnay swap tasting, where I got to see just how influential the winemakers' decisions are in the final wine product. And it made me realize just how important research is to allow our winemakers to make informative decisions to achieve the product they desire. My project focuses on ice wine research and specifically an ice wine yeast that is used in the industry. So my goal is to bioengineer an ice wine yeast strain to explore the roles of the 5 ALDs known as aldehyde dehydrogenase. These proteins are thought to play a role in acetic acid production. Ice wine juice has a high sugar content, which results from the grapes being naturally frozen on the vine and then pressed. So this high sugar content creates hyperosmotic stress to our yeast, which results in water loss. The yeast cells will then produce glycerol to draw this water back in, but this results in a redox imbalance. And the cells combat this redox imbalance by producing acetic acid. But acetic acid isn't really desirable in wine, as it is a volatile acid and impacts the quality of wine. So my goal is to explore which of the 5 ALD genes are responsible for this increase in acetic acid production. So I'm using CRISPR technology to knock out these genes in an industry wine yeast strain. And when I have successfully knocked out the yeast, the ALD gene in the yeast, I will then take it and perform fermentations so we can do biochemical analysis and determine concretely which ALD gene is responsible for this acetic acid production. So this is really essential preliminary research for researchers and winemakers so that we can know how the hyperosmotic stress of yeast influences and results in this increase in acetic acid production.